Hello everyone and welcome to today's webinar, Detecting Low Abundance Proteins in a Western blot, Overcoming Challenges for Improved Signal to Noise of Low Expressing and Precious Sample Targets. I'm Susie Valdez of LabRoots and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit them at www.thermofisher.com. Now let's get started. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Emily Halbrader, Product Manager, Protein and Cell Analysis at Thermo Fisher Scientific. For the complete biography on our speakers, please visit the Biography tab at the top of your screen. Emily, you can now begin your presentation. Welcome.
Thank you for that introduction. Just to recap again, today we'll be focusing on the detection of low abundance protein targets in a Western blot. Western blotting is a powerful and widely used protein research application as it allows the full visualization and characterization of target proteins in a complex mixture. An estimated 5 million Western blots are performed across the globe each year, which are utilized to help researchers better understand their individual protein of interest in highly popular areas of study, including drug discovery, cancer research, and disease. Western blotting is often performed downstream of common protein applications such as purification, protein-protein interaction, and gene knockout or gene silencing. It can also be used to determine if proteins are expressed, upregulated, or downregulated in response to external stimuli. Although Western blotting is well understood and very popular, it can often present challenges for certain proteins of interest. In particular, the detection of scarce proteins, whether it's a low abundant target or a low concentration of sample, can be a major challenge and often results in either faint or undetectable bands. This leads to repetition or re-optimizing the Western blot. Again, in this discussion, we'll focus on different methods and improvements that can be made to help Western blot detection of low abundant protein targets. The Western workflow involves steps from sample extraction through image capture and analysis. During sample prep, the protein is extracted from the source, whether from a cell, a tissue, or a subcellular fraction. Once extracted, the protein lysate is loaded onto the gel and run through protein electrophoresis to separate out the proteins by molecular weight. The gel is then placed into a sandwich-like structure to transfer out of the gel and onto a membrane for immunoblotting detection. Immunoblotting includes the washing, blocking, and antibody probing to detect a specific target of interest. Either chemiluminescence or fluorescence detection is then used to visualize the target. An imaging system or X-ray film can be used to then capture this final image and analyze the results. Each year, thousands of proteins are studied, and there's been an increasing need to detect lower amounts of protein target in the sample. Sensitivity is a growing need by many researchers in Western blotting. There are two primary reasons why sensitivity may be required. The first reason is that a very low abundant target is being detected in the Western blot. The target may be low abundant for a variety of reasons. For example, the protein concentration assay may indicate that the overall protein volume or concentration was lower than expected. With a lower protein concentration, and if limited sample is available, there may be a need to load smaller amounts of concentrated protein onto the gel. If the protein load is more dilute, a highly sensitive system may be needed to offset that limited sample and adequately detect the specific target. Another example is known low abundant targets. Usually this knowledge comes from experimentation or of known conditions. Proteins such as post-translational modifications, drug inhibition, and downregulation can lead to low expressed targets and therefore require higher sensitivity in detection. Finally, certain proteins are more difficult to extract and may result in lower yields of available protein. Membrane-bound or nuclear expressed proteins may be more sticky or stubborn during extraction and result in lower abundant targets in a Western blot. The other reason for sensitivity is in order to preserve precious sample. The sample source itself may be limited and then require effort to detect those proteins in a dilute sample. Certain tissues, such as human or mouse brain tissue, may be available in such limited quantities that it becomes necessary to only load a few micrograms onto the gel. Additionally, a single sample source may be needed over a variety of experiments, so that sample is therefore spread across these downstream applications. Whatever the individual reason is, the ability to preserve sample loads and still detect even the smallest amount of target protein is critical to making the right research conclusion in the Western blot. The Western workflow is highlighted here and again covers 6 key steps of how to improve the overall detection of low abundant targets. We'll be covering the factors that include efficient extraction and well-prepared protein samples, high-quality protein gel chemistries for effective protein separation, complete protein transfer from gel onto the membrane, the use of validated antibodies at optimal concentrations, increased low-end sensitivity with specialized reagents, and improving the dynamic range and detection capabilities with new imaging instrumentation. To improve the detection of low abundant targets, it's important to ensure efficient protein preparation to maximize yield and overall recovery of the protein target. There are a variety of different techniques for cell lysis, all of which work to break down the cell membrane to extract the proteins. When considering the best method of protein extraction for your experiment, it's critical to think about the sample source and location of the protein which you are probing for. The sample you are working with could be mammalian cell or tissue, bacteria, yeast, insect, or plant. Within that sample source, you may be extracting from specific locations, including either a membrane-bound protein, such as a cell receptor, a nuclear, or a cytosolic located protein. Wherever your protein is located, it's important to think about using optimized reagents for that specific target. These reagents should also be compatible with your downstream application, in this case, Western blotting, to preserve the protein activity and function. The table here illustrates the breadth of reagents that Thermo Fisher Scientific offers for high-quality, efficient, and specific protein extraction. The active ingredient in most of these lysis reagents are detergents, which help to disrupt the membrane and release the proteins inside. As an example, MPER, which is listed at the top of this table, is designed for complete cell lysis and contains a mild, non-denaturing detergent that dissolves cell membranes to extract and solubilize total protein from cellular compartments. To help ensure efficacy for research needs, we've validated MPER for use with several cell lines, including primary, suspension, and adherent cell types, as well as validation for Western blot applications. Homogeneous and efficient tissue protein extraction can be challenging due to the variety of different tissue compositions. fibrous tissues such as heart or kidney cannot be effectively extracted through freeze-thaw or manual methods without the aid of a detergent. Conversely, soft tissues like spleen or liver require a milder extraction protocol for optimal results. To help improve the overall extraction of tissue samples, TPER, also listed on this table, was designed and developed using a mild detergent that enhances the solubilization of proteins without compromising their function, and the neutral pH and salt concentration of this reagent enable a more efficient extraction from cell compartments to yield homogeneous lysates. While many of the reagents listed here can be used to better optimize extraction from the specific sample type and location, there are still several proteins that are not easily extracted. Inefficient extraction and low yield can present difficulties in detecting during the Western blot application. One such class of proteins that present inherent difficulties are plasma membranes called G protein-coupled receptors. These GPCRs are key to cell function as they play a primary role in transmitting signals across the cell membrane. They are the largest class of membrane proteins with greater than 1,000 identified and are a major drug target for genetic and immune system disorders. One of the major challenges in GPCR research is the ability to efficiently extract the receptors from the membrane without compromising their structure and functional integrity. In addition, GPCRs have poor solubility due to their hydrophobicity and are expressed at much lower levels compared to intracellular proteins. They often require enrichment prior to downstream analysis. As mentioned, GPCRs are difficult to extract, which often lead to harsher conditions that result in either denatured or unstable proteins at lower yields. They usually require further enrichment and stabilization of the target. Traditional protocols often use high salt or high pH buffers to disassociate the proteins from the cell membrane, but this risks damage to the protein integrity. Traditional methods are also laborious, time-consuming, and require expensive equipment. To address these concerns and issues, we recently introduced the Pierce GPCR extraction and stabilization reagent, an all-in-one solution to efficiently extract and stabilize GPCRs in conjunction. The reagents utilize a proprietary detergent solution where the sample is first harvested, washed, and then pelleted. The GPCRs are extracted using the GPCR extraction and stabilization reagent, followed by centrifugation. The resulting whole cell lysate contains solubilized and stabilized receptor proteins. The reagent has been tested for use with endogenous and overexpressed GPCRs, and receptor function has been verified by receptor ligand binding assays. In the data shown here, we can see the efficient extraction and solubilization of GPCRs from both cells and tissues using this new GPCR extraction and stabilization reagent. These are highlighted in column C in both examples. The data compares this new product to the Millipore transmembrane protein extraction kit, buffers A and buffer B, and to RipaBuffer, which is listed in column D. To perform this experiment, 3 GPCRs listed along the left-hand side were solubilized from HeLa HEK293 and mouse liver cells using the 4 different methods. The extracts were separated, transferred, and detected using SuperSignal substrates, and the images were captured using the iBright FL1000 imaging system. The GPCR extraction and stabilization reagent demonstrates improved banding and yield in the soluble fraction labeled S when compared to the insoluble pellet labeled P in both cells and tissue extracts. Using reagents like this can help better extract those target proteins for efficient detection in Western blotting applications. Moving into the next step of the Western blot workflow, It's important to choose an appropriate gel chemistry based on the molecular weight of the target and the expected load volume of the sample. Gel chemistries have been talked more and more as a key decision factor in optimal protein separation. There are a variety of gel options available, and it can often be overwhelming or even confusing to know which gel chemistry is the best one to use. When focusing on low abundant targets specifically, Bis-Tris gels can be an effective tool. For proteins of low abundance or for proteins with post-translational modifications, Bis-Tris gels help to provide improved band separation and overall detection quality. First off, Bis-Tris can help preserve the protein integrity within the sample. The neutral pH chemistry of the gel minimizes protein modifications or degradation that can cause poor band resolution. The result can be seen in the image to the right. In this example, the Bolt Bis-Tris gel shows the preservation of these full-length proteins listed across the top, while the Tris glycine gel from Biography shows multiple low molecular weight degraded protein fragments. In addition, Bis-Tris chemistries can help achieve superior band quality as they are designed to provide sharper, straighter bands. While we focus on Bis-Tris gels for the detection of low abundance targets, Tris glycine gels, which are a popular chemistry to be used, are certainly useful for detection of high abundant and broad range molecular protein targets. Detection of proteins, whether low or high abundance, is most efficient, clean, and optimal when the protein target is resolved in the middle portion of the gel. If your target is either high or low molecular weight, a broad-range gel is typically not effective at resolving these unique targets. To address these concerns, we suggest using the Tris acetate chemistry for high molecular weight targets and trisine for low molecular weight. These two chemistries are independently optimized to provide better separation for their given targets. In the image on the left, we can see the sensitivity improvements of the EGF receptor protein when using a Tris acetate gel compared to a broad-range Tris glycine gel. The 3 to 8% Tris acetate chemistry provides a matrix to allow larger molecular weight proteins to travel more effectively and separate for improved immunodetection. Conversely, the 16% trisine chemistry slows the migration so that low molecular weight proteins or peptides, which typically travel very rapidly through a gel, are better resolved. This can be seen on the left-hand side of the slide. Ion concentrations are also significantly higher at the bottom of a gel, and when used with low molecular weight proteins can cause disproportionately high current and transfer, leading to blow-through. Trisine gels keep these low molecular weight targets closer to the middle of the gel to reduce the risk of this blow-through happening. The example shown with trisine gels compared to Tris glycine gels separates and detects the cleaved caspase 3, shown on the right. The trisine gel is effective at separating out these two fragments of the target for better detection and sensitivity. Regardless of the chemistry used in your Western blot, it's critical to also consider the effective protein load volumes. If you see poor resolution or lane streaking, this can be a result of too much protein on the gel. As a general guideline, the maximum recommended sample load on a mini gel is between 10 and 15 micrograms of lysate per lane. When looking at low abundance targets, it's not uncommon to instinctively think that loading more protein will lead to higher probability of detection. However, this intuition is normal, but can actually jeopardize the quality of the blot and lead to suboptimal detection. While protein concentration is key to good band resolution, there are many instances where the overall concentration may actually be very low, either because the low yield of extraction or because the sample is precious and must be preserved. In these instances, when dilute sample is required, a higher volume is needed to be loaded on the gel to get adequate detection. The Wedgewell gel format allows greater overall volume of dilute samples to be accommodated without the spillover to neighboring wells. In the images below, we show trisglycine and Bis-Tris wedge well formats compared to Biorad gels. In both of these cases, we can see that loading up to 60 microliters of volume in the wedge well gel format does not have protein spillover or band smiling in the lanes compared to the non-wedge well formats. Wedge well gels are available in a variety of gel chemistries and again are ideal for loading high volumes of dilute samples in the detection of low abundant targets. Moving to transfer, we'll now cover some key tips for ensuring complete protein transfer from the gel to the membrane. To briefly review the transfer step, we can see the key format used in the schematic at the left. Transfer involves running a current through the gel membrane sandwich to move the proteins out of the gel and onto the membrane. A membrane is often used for detection as it is a stronger support compared to delicate gels and provides a surface on which proteins are more easily accessible. During the transfer, the SDS-bound negatively charged proteins migrate out of the gel and towards the positive anode. The membrane sits next to the gel and binds the proteins as they moved. Gel chemistries and transfer methods both play critical roles in the quality of the overall transfer. Focusing first on the gel chemistry, we can see in the image on the right how Bis-Tris gels compare to trisglycine gels when it comes to transfer efficiency and detection sensitivity. In these examples, A431 cell lysate extracts were electrophoresed and used on either the Bolt Bis-Tris or trisglycine gel. Both gel types were then transferred using the iBlot 2 from Invitrogen onto either nitrocellulose or PVDF membranes and then probed for the phospho-EGF receptor mouse monoclonal antibody, followed by an anti-mouse HRP secondary and detected with ECL. The Bis-Tris gel shows greater overall intensity and additional band detection at the low end, illustrating the higher and more complete protein transfer out of the gel and onto the membrane. Another important factor during transfer is the method and protocol used. Traditional wet tank transfer can provide high efficiency and is the widest used method. It relies on traditional tank formats with use of Tobin transfer buffers and manually setting up the transfer sandwich using an assortment of available membranes and filter papers. While wet tank transfer continues to be a very popular choice for many researchers, there are some downsides to this method, including the use and disposal of hazardous methanol-based buffers, longer amounts of time needed to complete the transfer, and performance risks such as blow-through or uneven transfer. Newer transfer systems have been developed to maintain and optimize transfer conditions while drastically improving the time of transfer to less than 10 minutes. Not only is convenience improved, but overall quality of the transfer can also be improved. This is critical when detecting low abundant proteins, as dead spots, blow-through, incomplete transfer, or blot-to-blot inconsistencies can result in inconclusive results. The Invitrogen fast transfer systems, which include our iBlot 2 and PowerBlotter devices, both come with ready-to-use stacks that offer performance advantages over traditional do-it-yourself stacks. Shown in the data here is a comparison of the ready-to-use stacks against a traditional self-assembled stack. Traditional stacks can lead to efficiency and reproducibility inconsistencies, resulting from uneven hydration of the membrane, insufficient removal of the buffers, or stack mishandling. Using too many or too few filter papers, incorrect stack orientation, drying of stacks, or contaminated buffers in the do-it-yourself stacks can cause problems. Using pre-assembled stacks with either the iBlot 2 or PowerBlotter can help eliminate the risk of these issues from occurring. In addition to improving the transfer consistency, fast transfer devices can also help improve sensitivity. While adequate transfer is possible across all transfer systems, it's often necessary to optimize and ensure good transfer on the system of choice. buffers, membrane types, and membrane binding capacities all play a role in affecting the transfer quality in a given system. In the data on the left, a lysate was loaded at decreasing levels to determine transfer efficiency across a range of concentrations. We can see an improved transfer at the low end of the lysate dilution range when we use the semi-dry power blotter system on the right compared to wet tank transfer. This is largely due to the optimized reagents and the optimized running program on the system. On top of the performance benefits, the transfer using semi-dry power blotter was accomplished in 7 minutes compared to a full hour using wet tank. Once the transfer is complete, the immunoblotting and processing steps begin. We'll start by discussing the importance of using validated antibodies to increase the sensitivity and quality for low abundant targets. At Thermo Fisher Scientific, we provide a large portfolio of available primary antibodies across thousands of target proteins, as well as a variety of secondary antibodies with either enzyme or fluorescent labels for Western blot detection. On top of the high-quality, robust portfolio offering, we provide validation and guarantee the performance of these antibodies in specific applications. In all Western blot experiments, but in particular when the protein of interest may be more difficult to detect or lower abundant, using a validated high-quality antibody is essential to maximize the quality of the result. The affinity of the primary for the target and its validated use in the specific assay application are both important considerations. With the antibody of choice, proper dilution should always be considered depending on the starting concentration of the antibody and the anticipated substrate that will be used. In a Western where higher sensitivity is required, it's common to consider using a higher concentration of antibody in an attempt to increase the sensitivity. However, high concentrations do not always mean better results. In fact, for low abundant targets, it is strongly recommended to use a higher sensitivity substrate and further dilute the primary and secondary antibodies. Too much primary or secondary, when used with these high sensitivity substrates, can oversaturate the blot or cause excessive background noise. These problems make it very difficult to effectively detect the target and make conclusive decisions on the data analysis. For primaries, a 1 to 1,000 to 1 to 5,000 dilution from a starting 1 mg per ml stock is a very typical first pass. However, if the blot is too saturated or not enough detection is acquired, the blot can be stripped and reprobed with a higher dilution of primary antibody. For secondary antibodies, it is highly useful to factor in the substrate of choice when determining the appropriate dilution. Again, with low abundant targets, a high sensitivity substrate should be used. These substrates are designed to give higher light emittance from the HRP enzyme label compared to standard or routine ECL substrates. When using these specialized substrate reagents, a higher dilution of antibodies should be used so as to prevent oversaturation of the blot. Instead of using a typical 1 to 5,000 or 1 to 10,000 secondary, a 1 to 100,000 or even up to 1 to 250,000 dilution of secondary can be prepared. The data shown in the middle of this slide illustrates oversaturation and optimal saturation in a Western blot. Too much signal, as seen on the top Western, makes it difficult to analyze, quantitate, or make conclusions. This often is the result when too much antibody is being used on the blot. Conversely, when properly diluted and optimized, the bottom blot shows a much easier to analyze Western across varying load conditions. In addition to using validated antibodies at optimal dilutions, using detection reagents specifically designed to increase sensitivity can help improve detection of low abundance targets. Chemiluminescent substrates continue to be the most commonly used detection method for Western blotting due to their flexibility across film and imaging systems and the ability to provide higher levels of sensitivity. However, low abundant targets can still result in detection challenges if the right substrate and optimizations aren't incorporated. Common problems include weak or no signal, poor signal to noise, or wasted sample and reagents if repetition is required. These challenges are most often experienced when working with low abundant targets. In those instances, using high sensitivity substrates can greatly increase the chance of adequate signal. Thermo Fisher Scientific most recently introduced an improved high sensitivity substrate that provides the greater levels of detection sensitivity. The SuperSignal West Atto substrate provides ultimate detection sensitivity designed specifically for detecting very dilute or low abundant targets. It can provide over 3 times greater sensitivity than other conventional high sensitivity substrates and detects proteins where routine substrates fall short. Further, it has the greatest level of signal to noise to allow users to visualize bands where previously they were undetectable. An example of this advantage is shown in the 2 Western blot examples in the top right corner. SuperSignal West Atto presents a unique value in allowing users to also save on their sample or antibodies. We'll cover these attributes in greater depth in the following 2 slides. In this data comparison, we can see how SuperSignal West Atto compares to other substrates that are marketed for high sensitivity Western blots. In the blots and graphs shown, HeLa lysates were serial diluted and loaded at the indicated volumes. All blots were transferred and probed for AKT and captured on the iBright imager at the same exposure times. When we compare using SuperSignal West Atto to substrates provided by Biorad, GE, or Millipore, we can see the increased overall intensity and signal above background advantages that SuperSignal West Atto can provide. This signal is especially important in Western blots looking to detect very low amounts of target protein or very dilute samples. To further illustrate how SuperSignal West Atto can help preserve precious sample, we can look at the case study here. In this experiment, we can compare SuperSignal West Atto to routine substrates from Biorad and Millipore. Routine substrates are often used for medium to high abundant protein targets or routine Western blots. However, even if a protein target is medium to high abundant, the sample itself may be precious and requires as much conservation as possible. Samples such as tissues or human cell extracts may be very limited in availability or, again, used across several experiments, so each microgram used is considered precious. If routine substrates are typically used, this usually requires higher loads, upwards of 10 to 20 micrograms, to get the detectable signal that's needed. This is illustrated in the yellow and blue boxes and corresponding charts. However, SuperSignal West Atto can be used in replacement of routine substrates to help preserve sample, and in fact load up to 1/16 of the amount compared to routine substrates and produce the same signal detection. This is illustrated by the red box and corresponding chart. When samples are so precious and only a couple of micrograms or less are available, SuperSignal West Atto can provide confidence in obtaining adequate detection sensitivity in these cases. Additional reagents to improve signal detection include blocking buffers. When poor signal to noise is obtained on a Western blot, a contributing factor is often the blocking buffer used. While dry milk is a common choice and works for many Western blot needs, this blocking buffer contains a variety of proteins that are not well defined or controlled and can lead to non-uniform and suboptimal blocking efficiency. Using a purified blocking buffer like the Thermo Fisher Scientific Pierce Clear Milk Blocker or Starting Block Blocking Buffer can help improve the signal to noise and enhance the sensitivity in your Western blot. An example of this low-end detection improvement is illustrated in the Western blot shown at the right. All 3 of these Western blots contain equivalent serial dilutions of HeLa lysate. Once transferred, the blots were blocked with the respective blocking buffers, starting block, clear milk, or 5% non-fat milk. They were then simultaneously probed for IRAC1 and BCL-XL. SuperSignal West Atto substrate was used to detect the low levels of protein targets on all three blots. As demonstrated, starting block provides the greatest level of sensitivity and overall signal-to-noise improvement, followed by the Pierce clear milk. Non-fat dry milk reduces the overall signal intensity and is not able to provide detectable signal to the same sensitivity level as the other blocking buffers used. When ultimate sensitivity is required, it can be very helpful to use specialized blocking buffers like starting block, designed to increase sensitivity and signal to noise. A unique and exceptional tool for signal enhancement is the SuperSignal Western blot enhancer, which is used to help enhance the detection of low abundant or weakly immunoreactive antigens by increasing sensitivity up to 10-fold. The SuperSignal Western blot enhancer is an innovative technology that works to increase the specific binding of the primary antibody to the target protein, while also reducing the overall background of the blot. The reagent is a two-part system, first containing a membrane treatment that helps to decrease the background and nonspecific binding sites. The second reagent works as a primary antibody diluent to increase the exposure of the antibody epitope binding sites and promote effective targeted binding to the protein of interest. This improved specificity is especially important for poor quality or low affinity antibodies. The overall result using the enhancer is a cleaner, more sensitive Western blot detection, as illustrated in the blots on the right-hand side. When using the SuperSignal Western blot enhancer, detection sensitivity can be increased up to 10-fold, and the overall signal above background is greatly enhanced. Moving into the last step of the Western workflow, we end at the image capture and analysis step. Using modern technologies such as CCD imaging systems can greatly improve dynamic range and low-end detection. The two imaging modes most commonly used in Western blotting are either film or CCD cameras. Film capture is the traditional method. While relatively straightforward, it presents its challenges as optimal exposure times may be more difficult to obtain and require repetition. Darkrooms can be expensive and more difficult to maintain. Conversely, CCD imaging is a fast-growing technology that is being used more widely and adopted by Western blot researchers. It is a modern technology that can be used across a broad range of applications, including both chemiluminescent and fluorescent Western blotting, DNA gel staining, and protein gel staining. The instrument and software enable easier data acquisition and optimal exposures as they use smart features to predict the best exposure times for the light that is being generated on the blot. However, these imagers do require an investment, training, and a designated space within the lab. Despite these factors, many research labs are finding significant value in CCD imaging systems. The Invitrogen iBright imaging system is a technically advanced yet easy-to-use system for Western blots. It enables a broad range of capabilities for Western blotting, protein gels, and DNA gels, and contains a powerful camera for high-resolution images. The innovative smart interface makes it an easily adaptable platform that brings the benefits of imaging systems without excessive training. One of the biggest benefits to CCD imaging is the expanded dynamic range and the improved image capture. CCD imaging allows for multiple images to be taken and stored, so it's easier to achieve the optimal exposure time the first try. In the iBright system, the camera uses a smart exposure feature which helps to predict the best exposure time for the blot. The data here shows blots for 3 different targets and at 10-second exposures. Either the iBright imaging system or traditional film are used to capture the image. When using film, the blot oversaturates at the top end very quickly compared to the iBright imager. In addition, the iBright is able to maintain the low-end sensitivity and provide a linear detectable range far greater than film, which is shown in the graph to the right. Additional supporting data of the signal linearity generated with iBright is shown here. While Western blot's image with X-ray film may appear to have a linear detectable range, when performing densitometry on the individual bands, it's clear to see that the signal captured on film has a much narrower dynamic range compared to signal captured with the iBright imaging system. Images acquired on the iBright show a linear detection range for PDI from 125 nanograms up to 1,500 nanograms of helolysate, whereas with the film, the linear signal is only from 125 nanograms to 500 nanograms. In summary of today's review, we covered a variety of techniques and products that can help improve signal detection and sensitivity of low abundant targets across the Western workflow. These considerations include efficient protein extraction and optimized target-specific reagents, use of gel chemistries that improve protein separation and preserve protein integrity, complete and efficient transfers through the use of optimized gels and transfer methods, ensuring the use of validated antibodies specific for Western blotting, incorporating high sensitivity detection reagents such as SuperSignal West Atto and SuperSignal Western Blot Enhancer, and the use of CCD imaging systems for improved dynamic range and image exposure. Western blotting is a highly popular technique and an essential research tool for protein studies. We hope these tools can be useful in your Western blot optimization and troubleshooting to achieve successful and efficient Western blots, particularly for low abundant targets. I'll now open up for any questions.
Thank you, Emily, for that informative presentation. And we will now start the Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So let's get started. Our first question, Emily, from an audience member is, if a routine ECL substrate is used first and no bands are seen, can the SuperSignal Atto substrate be used directly after, or is stripping and reprobing necessary?
So that's a great question. Typically, if you're using an ECL substrate and you're not seeing any bands, we would recommend stripping off the antibody, so using a stripping, something like a restore stripping buffer to remove those antibodies. And the reason for that is because the antibodies may not be fully optimized for the higher sensitivity substrate, it's best to remove those and re-optimize. That being said, we do know quite a few customers prefer to move up the line of substrates, if you will. So if ECL doesn't show a detection, they may add a stronger sensitivity substrate. So you can certainly try this, and then once you're able to detect that signal, it is best to then strip and then re-optimize using properly diluted antibody concentrations.
Thank you. And Emily, can long film exposures be used to increase sensitivity when bands are not detectable?
So exposure time is a great way to influence the sensitivity, and many researchers do use varying exposure times to help increase that sensitivity. So when you're using film, yes, you can certainly expose for a longer time. The caveat is often that longer exposure times can start to increase the background, but they can help get that signal detected. So whether you're using film or even an imaging system like the iBright Imager, a longer exposure time can be useful. Again, the nice thing with iBright imaging systems is that smart exposure feature can be used to help predict the optimal exposure time. So it may help you predict that 15 or 30-minute or even longer exposure to get the sensitivity of your particular Western blot.
Thank you for that. And I want to also thank our audience members for their great questions coming in. Our next question from an audience is, if using higher sensitivity substrate in a system where non-specific bands are detected, won't that non-specific detection increase significantly as well?
So non-specific bands are certainly problematic for a lot of researchers. In theory, yes, if you're having issues with non-specific bands, increasing the sensitivity, that signal overall on the Western blot is going to also be increased. So what I would suggest if non-specific bands are a problem, there's a couple of things to look at. The first is definitely the primary antibody. So if the antibody that you're using is less specific to the target, considering a different antibody, and especially an antibody that's validated for Western blotting, would be the first step to evaluate. The other product that you can use that we discussed today is that SuperSignal Western blot enhancer. And again, what that enhancer reagent does is it's a two-part system, and the second reagent that's used is an antibody diluent, and that diluent helps to increase the specific binding and the efficacy of the antibody to target binding, and so can actually help reduce non-specific binding in the system. So a combination of looking at the primary antibody, using one that's validated and very specific for the target, and potentially incorporating an additional reagent like the enhancer reagent can help reduce that non-specific binding so that your sensitivity, which you might need to be improved, is improved only for your specific target.
Thank you for that. Now, how can you increase sensitivity of chemiluminescent blots that are stripped and reprobed?
So with stripping and reprobing, the key there is to fully remove the antibodies that were used for immunoblotting in the first detection. So we have two reagents that are used for stripping and reprobing, and those reagents are Restore and Restore Plus. To get the best sensitivity of both targets, specifically if you're using antibodies that are very specific for the target, the Restore Plus is a great reagent to use because Restore Plus is designed to fully remove antibodies, especially antibodies that are very specific to the target and therefore very tightly bound on the membrane. So if you use Restore Plus, that reagent can help fully remove and strip away the antibodies that were used in the first detection, and then reprobe with the new set of antibodies for the other target or targets. So using Restore Plus is a great way to increase the sensitivity during that stripping and reprobing process.
Thank you. And Emily, how about Femto and Pico? How sensitive are they compared to Atto?
So that's a great question, and it sounds like very familiar with the SuperSignal substrate. So the SuperSignal Pico or Pico Plus is really designed to be more of a routine ECL substrate. So better used when your target is either medium or high abundant, and you have plenty of sample to load onto the gel. Femto is also a high sensitivity substrate. When compared to West Atto, SuperSignal West Atto is going to be even more sensitive than Femto, again by about threefold more sensitive. So using West Atto, it comes to be very important when that sample availability is very low. So if you only have a couple of micrograms, West Atto is going to be able to do even a better job than SuperSignal West Femto is in detecting those targets. So West Femto, if you're using that today, that is continued to be available, but if you need even more sensitivity, or if you have a limited sample to load onto the gel, the new SuperSignal Atto can be used to increase that sensitivity even more than Femto.
Thank you, Emily, and thank you for your time today and your important research. Would you like to provide any closing remarks for our audience today?
Well, I just want to say I appreciate everyone's time to listen into this webinar. We certainly hope to continue to be a support here at Thermo Fisher Scientific for research needs, and we will look forward to any other questions that come in even after this webinar. We will certainly reach out and support any further needs. So thank you very much.
And thank you again. And we'd like to also thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. And before we go, I want to remind the audience, thank them for joining us today, and also for your interesting questions. Like Emily said, questions we did not have time for today, and those submitted during the on-demand period, we will address them through the speaker via the contact information you provided at the time of registration. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, bye-bye.